The function of TET enzymes in DNA demethylation and gene regulation. DNA methylation has long been recognized as a fundamental epigenetic mechanism regulating gene expression and cellular identity. The methylation of cytosine residues, predominantly at CpG dinucleotides, has traditionally been associated with gene silencing, particularly when occurring in promoter regions. However, the dynamic nature of DNA methylation patterns across developmental stages and in response to environmental cues necessitates an equally dynamic mechanism of demethylation. This is where the 1011 translocation TET family of enzymes plays a crucial role. TET enzymes catalyze the stepwise oxidation of 5-methylcytosine, 5-MC, to facilitate DNA demethylation, thereby influencing gene regulation, cell differentiation, and disease pathogenesis. In this documentary, we will explore the function of TET enzymes in DNA demethylation and gene regulation, drawing on extensive research, experimental evidence, and clinical insights to elucidate their pivotal role in epigenetic modulation. The discovery of TET enzymes can be traced back to the identification of TD1 as a fusion partner in acute myeloid leukemia, involving chromosome translocations. Subsequent research revealed that TT1, along with TT2 and TT3, belong to a family of dioxygenases dependent on iron and alpha-ketoglutarate for their enzymatic activity. Structurally, TET enzymes share a conserved catalytic domain containing a double-stranded beta helix fold, which is crucial for their interaction with DNA and the oxidative conversion of 5-MC into its derivatives. The demethylation process catalyzed by TET enzymes occurs through a stepwise oxidation of 5-MC to 5-hydroxymethylcytosine, followed by further oxidation to 5-formylcytosine and 5-carboxylcytosine. These oxidized derivatives can subsequently be removed through base excision repair or passive demethylation during DNA replication if the maintenance methyltransferase DNMT1 fails to recognize these modified cytosines. The identification of 5-HMC as a stable intermediate raised significant interest, as this molecule is enriched in gene bodies and enhancers, suggesting a role beyond demethylation. Genome-wide studies have revealed that 5-HMC is highly abundant in neuronal tissues, embryonic stem cells, and germ cells, indicating its potential function in maintaining cellular plasticity and regulating gene expression. TET enzymes play a crucial role during early embryonic development when epigenetic reprogramming occurs on a large scale. Global DNA demethylation in the zygote involves the activity of TET3, which converts paternal 5-MC to 5-HMC, while the maternal genome undergoes passive demethylation. Studies in mice have demonstrated that TT1 and TET2 are indispensable for the proper establishment of pluripotency and lineage specification. In human embryonic stem cells, TET1 and TET2 have been shown to maintain pluripotency by regulating genes such as NANOG, SOX2, and OCT4. Experimental data suggest that loss of TET activity results in the hypermethylation of these promoters, leading to reduced expression and compromised self-renewal capacity. Furthermore, TET enzymes are critical during the transition from naive to primed pluripotency, where the dynamic regulation of 5-HMC at enhancer regions facilitates lineage-specific gene activation. The brain exhibits the highest levels of 5-HMC among all tissues, implicating TET enzymes in neuronal development and function. TET1 and TET3 are particularly abundant in neural progenitors, where they regulate genes involved in synaptic plasticity, learning, and memory. Genome-wide mapping studies have identified 5-HMC enrichment at active enhancers and promoters of genes critical for neuronal differentiation, such as BDNF, or brain-derived neurotrophic factor, and NeuroD1. Mouse models with TET1 depletion have shown deficits in memory formation and synaptic plasticity, underscoring the enzyme's importance in cognitive processes. Additionally, TET enzymes have been implicated in activity-dependent DNA demethylation in mature neurons, suggesting a mechanism by which environmental stimuli induce long-term changes in gene expression. For instance, exposure to enriched environments or learning paradigms results in increased 5-HMC levels at genes associated with synaptic remodeling and neuronal connectivity. Hematopoietic stem cells, or HSCs, rely heavily on TET2 for normal differentiation and self-renewal. Loss-of-function mutations in the TET2 gene have been frequently observed in hematological malignancies, including myelodysplastic syndromes, chronic myelomonocytic leukemia, and acute myeloid leukemia. These mutations lead to aberrant DNA hypermethylation, which disrupts the expression of genes involved in differentiation and apoptosis. Mechanistic studies have shown that TET2 modulates the methylation status of enhancer regions controlling myeloid lineage commitment. For instance, the hypermethylation of the PU1 enhancer, a critical transcription factor for myeloid differentiation, correlates with the impaired differentiation observed in TET2 mutant leukemia cells. Furthermore, TET enzymes regulate immune cell function by modulating cytokine gene expression and promoting T-cell differentiation into regulatory T cells, which are essential for immune tolerance. The involvement of TET enzymes in cancer is complex, with context-dependent roles in tumor suppression and oncogenesis. TET2 mutations, as mentioned, are strongly associated with hematologic malignancies. In solid tumors, altered 5-HMC levels have been detected in cancers such as glioblastoma, melanoma, and colorectal cancer. Decreased TET activity leads to the hypermethylation of tumor suppressor genes, contributing to uncontrolled proliferation and metastasis. Conversely, TET enzymes can also facilitate oncogenic transcriptional programs. In certain breast cancers, increased TET1 expression has been linked to the activation of metastasis-promoting genes through 5-HMC enrichment at their enhancers. This duality suggests that TET enzymes act within a broader epigenetic context influenced by cellular identity, environmental factors, and oncogenic signaling pathways. Epigenetic modifications, including DNA methylation and hydroxymethylation, are susceptible to environmental influences. Factors such as diet, stress, toxins, and aging have been shown to modulate TET enzyme activity. For instance, alpha-KG, a key cofactor for TET enzymes, is produced through the tricarboxylic acid cycle, linking cellular metabolism to epigenetic regulation. Caloric restriction and exercise, which enhance mitochondrial function, have been associated with increased TET activity and 5-hydroxymethylcytosine levels in multiple tissues. Exposure to environmental toxins, such as bisphenol A and heavy metals, can impair TET function by altering cellular redox states or depleting essential cofactors like ferrous iron. In a study involving mice exposed to lead during development, researchers observed a significant decrease in hippocampal hydroxymethylcytosine levels, correlating with cognitive deficits and altered gene expression profiles. Aging is accompanied by widespread epigenetic remodeling, including changes in DNA methylation and hydroxymethylation patterns. TET enzymes have been implicated in age-related cognitive decline in neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease. Post-mortem analyses of Alzheimer's disease brains have revealed reduced hydroxymethylcytosine levels at genes involved in synaptic transmission and memory-related processes. Experimental models suggest that TET-mediated demethylation may mitigate neurodegeneration by promoting the expression of genes associated with antioxidant defenses and neuronal repair. Additionally, increased oxidative stress during aging can impair TET activity, thereby disrupting the epigenetic regulation of genes critical for brain function. The therapeutic potential of targeting TET enzymes in disease contexts is gaining traction. In cancer, demethylating agents such as 5-azacitidine and decitabine, which indirectly influence TET activity, are currently used in the treatment of MDS and AML. The development of more selective modulators of TET activity, potentially through the manipulation of metabolic pathways supplying alpha-Kg, presents an exciting avenue for cancer therapy. In neurodegenerative diseases, strategies aimed at restoring TET function and 5-HMC levels may hold promise in preserving cognitive function. Preclinical studies have demonstrated that dietary supplementation with alpha-Kg or ascorbate can enhance TET activity and partially reverse age-related DNA methylation changes. TET enzymes occupy a central position in the dynamic regulation of DNA methylation, influencing gene expression during development, cellular differentiation, and disease progression. Their ability to mediate DNA demethylation through the oxidation of 5-MC to 5-HMC and subsequent derivatives has reshaped our understanding of epigenetic plasticity. From embryogenesis to neuronal plasticity, immune regulation, and cancer, TET enzymes emerge as critical modulators of genomic information beyond the DNA sequence itself. Ongoing research continues to unravel the complexity of TET-mediated epigenetic regulation, offering new insights into cellular identity, disease mechanisms, and potential therapeutic interventions. As we advance our understanding, the intricate interplay between TET enzymes, environmental factors, and cellular metabolism will undoubtedly remain a focal point in the evolving field of epigenetics.